Oh, hello, everyone. I'm Marc Rosenzweig from West Bloomfield, Michigan. I'm an outpatient and I'm going to my 11th year. And anything else, Summer, I missed out on? Without Summer, I don't know what I would do. So I'm going to go ahead and launch the first question.
When were you diagnosed?
Thanks, Summer.
Do we have the results here? When were you diagnosed? We have 25% of us, 25% of us 3 to 4 years, 19% of us 1 to 2 years, 15% of us 2 to 3 years, 8 years or longer are 10%. Did I do 12% under a year? I should just really go right down. 10% are 4 to 5 years, and 8% of us are 6 to 7 years. Thank you, Marc. And what is the type of treatment you are currently receiving? Okay, here we go. I'm going to try a different approach this time. 1% are chemo plus a TKI. 59% of you are on alectinib. 7% are on brigatinib, Yvonne and I included. Lorlatinib, 27%. Chemo, we don't have any straight up chemo people. So 0% for that. And 4% of you are on a clinical trial. Thanks, Marc.
And some radiation. Targeted radiation.
Okay, we're going to move
Target
forward.
radiation.
And I'm going to just pull up one. I'm going to introduce Dana Credo, and she's going to explain to us our healing arts programs.
Hi, everybody. So this week is a little bit lighter in our healing arts programs. We have chair yoga, which is on a separate Zoom channel. Then we have new alkies at 9 on Wednesday. On Thursday, we have art therapy at 1 Eastern and care partner check-in at 9 p.m. And then book club on Friday at 1 p.m. This is a great opportunity to, some of these, it's talking about, talking with people who understand, the troubles you're having with your medication, or you just want to not feel alone. In other words, other groups you don't have to talk about cancer at all, like our book club. Just come meet other people so you never have to feel alone. And, if you can come ask us anything, with people who run the groups, and if we don't have the answer, we'll find the answer for you.
If you have any questions, they're all ALKtALK at ALKpositive.org. And I'm going to pass the mic to Summer.
Thank you all again for joining us this evening. My name is Summer Farman. I am a patient diagnosed in 2020. I've been helping Marc with ALKtALK since 2021 as the ALKtALK program coordinator. We are here to educate, inspire, empower all of you to make decisions in your care, you or whoever you're here to support. So thank you for joining us. We find it's important to educate you all too on what ALK Positive Inc. is doing behind the scenes. We are a patient-driven organization, a registered charity with a goal to extend and enhance the quality of life and life expectancy of ALK positive patients around the world. And we are laser focused on doing that. A very specific portion of that is those medical committees working tirelessly behind the scenes. So we have Dr. Ken Culver here, our Director of Research and Clinical Affairs, and Dr. Marc Muskavitch, a patient and also overqualified medical committee member. And then staying up from London is fellow board member Yvonne Diaz, here to help moderate this talk with Marc and I tonight. So thank you all so much for joining us. And Ken, take it away.
Thank you, Summer, Yvonne, everyone in ALK Positive. It's a pleasure for Marc and I to be here. And it's a thrill to be able to tell you about some of the exciting things that we've been doing and how we've been investing time and money into things that we think are really going to be beneficial to the ALK patient community. There we go. So today I want to talk about your ALK positive medical committees. And when I arrived about 1.5 years ago, there were already medical committees set up and doing lots of great work. And one of the reasons why ALK Positive hired me is we wanted to try to further accelerate and expand your treatment opportunities. So all that we're doing is about accelerating and improving and enhancing and getting more treatment opportunities, because while you're all ALK patients, each of your tumors is different, and we need to take that in consideration in how these new therapies progress. So the medical committees of ALK Positive have about 30 patients, and caregivers who were involved. And some of those, 2 of the committees meet every week, and 1 of them meets every other week. So we're getting together on a very regular basis. This is the mission from our RAC, our Research Acceleration Committee, but I think it captures in one phrase what the goal of our medical committees are at large. We want to motivate, facilitate, and enable the development of new commercial therapies. And commercial is in there because once they get on the market, they become available to many, many more people. And we want to do that for therapies and diagnostics that benefit across the ALK positive patient community. So what do companies need to know about ALK positive to be able to commercialize something for you? Well, there are 5 things that we really have to be clear about. One is, what is ALK positive lung cancer? Most of them don't know, even though they may be trained in medicine and science. 2, what's the patient unmet need? A lot of people think, oh, there's a lot of great TKIs for ALK. Why do we need to be investing in that? 3, what's the clinical rationale? There are a lot of really good clinical reasons why ALK patients are an ideal place for companies to develop drugs. 3, what's the scientific justification? What is it that's going on in ALK positive cells that makes it a logical way for them to use their drug? And 5th, these companies have to have a business case. Is ALK a rare disease? If so, how can I make any money? We just have to be honest. That is a question that's being asked behind the scenes. So what kind of progress are we making towards these 5 things? And I'm going to go through and show you on those 5 things how we're talking to companies. But I wanted you to know that in the last 1.5 years, we've started our own internal research group, and Marc's going to talk about that. Then the goal is to find new targets in ALK tumor cells. And already some of the data that we've generated, and it's all been generated by bioinformatics, no laboratory work, has already contributed to a new trial that's in development from a vaccine company. 2, we have several companies have already recognized the value of ALK lung cancer trials, and they're already planning to start trials in ALK positive. Would have never happened if the ALK Medical Committee hadn't been engaging, pushing, teaching, educating, motivating, all those things. Some of those, of course, are going to require our financial support, because when these small companies they planned for other cancers. And then we came by and they said, oh, ALK's really important. We should add it, but we don't have the money. And so in some cases, it will require some financial support. And we just want you to know that being able to motivate, facilitate, and enable takes also money in some cases. And then 3, our grantees, we've awarded a number of grants through LCRF and longevity, and those grantees we continue to track, and they're working hard and continue to investigate mechanisms of TKI resistance and searching for new therapeutic options. So there are really important, tangible things that are coming out of the work of the medical committee. Now, here's another sort of benchmark for what we've been doing. We are currently following 82 companies who we believe have drugs in development that could be applicable to ALK patients. And we've already met with 10 biotechs just in the last almost 4 months. And the asterisk means we have ongoing discussions with them. 11 pharma, 18 universities, 3 major health systems, and 5 other organizations that are important to getting new ALK therapies out there. So we have been really busy. So we're having multiple discussions a week. And I've been fortunate to be able to travel to some of the lung cancer meetings where I can meet new people, particularly from these companies, and be able to continue those discussions and advocating for the ALK community. So how is it that we try to motivate and facilitate the development of new drugs? And it'd be really good so that when you're talking to friends, coworkers, family, and you're trying to explain what it is why ALK is so important, not just for you, but for others in this community, here are the key points that we try to make to give the business case, the clinical case, the science case, and the patient case. So what are the unmet needs in the scope of ALK positive? So something we all know, right? TKIs have improved survival, and the survival for ALK patients is longer than any other subgroup of lung cancer. But they're not curative, and we know that. And our goal is to make therapies, bring in therapies that are curative. But we know that once resistance develops to all the TKIs, it becomes very complicated. And there are a whole lot of different ways people treat it, including chemotherapy and clinical trials. And we're trying to really enhance the clinical trial options. This is a really, really important point, and I'm going to highlight this several times in my presentation. The ALK positive patient community is bigger than most people realize. So let's think about it first from an incidence point of view. If you say 5% of the 200,000 new cases a year are ALK positive lung cancer, then that means there's 10,000 new patients every year that are diagnosed with ALK positive lung cancer. And since there are about 10 times more lung cancer patients globally, that brings it almost to 100,000. But incidence only tells you the number of new cases in a defined period of time, in this case, 1 year. But what matters, I think, even more is what's the prevalence? In other words, how many people are living with ALK positive lung cancer in the United States? To get from incidence to prevalence, you multiply times the median survival. So in the old days in lung cancer, the incidence and the prevalence were pretty close to one another. But in the case of ALK positive, the prevalence is probably about 70,000 people in the US and almost 10 times that globally. So that means ALK positive lung cancer is not rare. And I think one of the key messages I want you to take home when you talk about ALK positive, I would recommend you say there are tens of thousands of people in the US who have ALK positive lung cancer, because when we call it rare, people get the impression that it's somehow less important, less of a public health interest. And I think that's wrong. About the business case. So if you add together, Jeff Sturm did this, if you add together the revenues made by Takeda, Genentech, and Pfizer, it's now probably about $3 billion a year. And when we show these numbers to the biotechs out there, they're blown away because they had no idea that they thought ALK was taking care of with TKIs. They thought ALK was rare. Therefore, what money could you make? And so I think it's practically speaking, here you can see there's a really good business case and a really important need that should be addressed by companies developing new therapies. So another thing that is an issue that really has to have attention is that ALK positive lung cancer patients are commonly included in the oncogenic driver cohort. So they include EGFR, ALK, ROS, RET, HER2, BRAF and so forth in one cohort. And I'm going to show you why that's really doing a disservice to the ALK community, to all those communities for that matter, because they're not all the same. Even all ALK lung cancers aren't the same, and you get way more diversity when you start throwing in other cancers whose primary driver is different. And what happens is you then end up with clinical trials that have a lot of EGFR patients, but only 2 or 3 ALK and maybe 1 ROS. You can't draw conclusions on 3 patients on whether that therapy works. You might get excited, you might be disappointed, but those impressions could both be wrong. And so this is one of the key points we're making, where we need to make sure that if you're going to include ALK patients, please enroll enough patients so we can know whether or not it's benefiting ALK patients or not. Immunotherapies. So we've been saddled in the ALK community with, well, immunotherapies don't work. They don't work. There's been 3 studies that show that if you give Keytruda or you give Opdivo, that it doesn't work. And I think what's really important here, and we all realize, is that studies with PD-1 and PD-L1 inhibitors have only shown a limited activity, but they're only one of many kinds of checkpoint inhibitors. And I'm going to come back to that. And so we don't throw the baby out with the bathwater here. We say, okay, that checkpoint didn't work. What about the other 15? I'm going to come back to that. Now, on this point of being rare, you see on the top of my screen here, this was a slide presented let's see, when was I there? It was presented yesterday morning when I was in Texas at the Texas Lung Cancer Conference. But it was from Alex Drillin, who is at Memorial Sloan Kettering in New York, and he was giving a presentation on RET and ROS, rare kinds of lung cancer, of which ALK is not one. And he was saying that as lung cancer is a common cancer, rare subsets are not uncommon. He's saying here that there's at least 2,000 patients a year that are incidents, right? So the number of new cases in a year for RET and ROS together is 2,000, right? That's 20% of ALK. That's not rare in his mind. So we certainly aren't rare when it comes. But then here was his next slide. Calling a cancer rare may lead to de-emphasis of optimal testing strategy. If doctors think, well, it's rare, it's unlikely for people to have it, therefore, why bother? The EGFR is negative, okay, then we'll just give them chemotherapy and immunotherapy. There's a lot of different rationales. And what that also means is that people don't get the right testing. They don't get the optimal testing strategy, which in this case should be a DNA-based test to look for ALK. And if negative, there should be an RNA test to confirm, because it's much more sensitive at finding fusions. We want really to know, when you talk to other people, the question is, did you get a DNA test and did you get an RNA test on your lung cancer specimen to know for sure that you don't have ALK? Now, what about this idea that the different subtypes of lung cancer are similar but different? And keep in mind, this is a presentation we're giving to biotech and pharma companies. And we're really trying to make the point that just compare ALK and EGFR. In this particular case, they're looking at 74 ALK and 374 EGFR in this particular paper. What they found is the median age was much lower in the ALK. We all know that the smoking incidence, non-smoking was much higher than in EGFR, and the performance status, the relative health of the person at diagnosis, was lower for ALK. So if you're a physician or a patient or a nurse and you're looking at this, you're saying, well, those look like different populations. But now you apply a TKI to those populations, and you see the red, which is ALK. You see the green, which is the red, which is ALK with more than one TKI. The green is ALK with one TKI. The blue is EGFR, and the black are the wild type. So the point of this slide is the following. If you lump these 2 different kinds of subtypes together, you're going to confound your results, because you're really enrolling 2 completely different kinds of cancer. It also can produce underpowered data, right? You can look at this here and say, well, that the ALK group is small relative to the EGFR, and that could really lead to a delay in getting patients, ALK patients, a drug that works, because there aren't enough ALK patients, so you don't really know. So then the company doesn't invest in it, or ALK patients are concerned that, maybe there's a better option, and it all becomes, I think, something we want to avoid. So we care about the EGFR and other oncodriver groups. We want everybody to get a fair test of the therapies in those subpopulations, not some kind of throw everybody together and see what falls out. Now, what about this question of immunotherapy? So I mentioned earlier, there have been 3 studies that showed that these PD-1 and PD-L1 inhibitors don't have much activity in ALK. And this is from Justin Gaynor's paper in 2016. And this is the EGFR and ALK cohort. Here they did separate cohorts and then they lumped them together. But it really didn't. We've all heard this, and there were 2 other studies that confirmed it. But as I mentioned before, this is only one kind of checkpoint. And for those of you who might not be familiar, checkpoints are proteins on T cells that turn the immune system on or off. So PD-L1 and PD-1 expression turn T cells off. So if you give that inhibitor of those, now T cells can be activated. And that doesn't work for everybody in lung cancer. It's not just ALK. But when you ask yourselves, well, how many of these checkpoints could there be? There are a lot. And right here in this representation, looking at the surface of a T cell, there are 16 different checkpoints. So why would you say, well, PD-L1 didn't work, that therefore the rest of these won't work? And so what we say to companies is, okay, we all agree, no one's seen activity with single agent PD-L1 checkpoint inhibitors, but what about all these others? They should be tested, and we're actively pushing immunotherapy along, and we've got data to support our assertion, and it comes in this slide. So, we funded, we ALK Positive, Dr. Chiarli to do some work, and Dr. Awad, and this is from their animal experiments with their vaccine. So when you look at a graph like this, where you see the lines coming straight down like this, you look at the axis and you'll say, this is a time after they started the therapy, and this is how many of the animals are surviving. So in this black line, all the animals were dead in 25 days. And so if you look over here, that's the untreated group. Now this cell line, if you look now at the purple and the dark blue, they lived out to 50 days. Well, that was lorlatinib, and lorlatinib plus a PD-1 inhibitor, which we already know, right, doesn't in humans doesn't have a lot of activity. Now we look at this light green, yellow, gold, whatever color it is, it goes out to almost 200 days. That's lorlatinib plus a different checkpoint inhibitor called CTLA-4. Now you go to the green, a red, and then let's go all the way up here. In this case, the animals are out getting close to a year. They didn't develop brain mets. All of them were alive. And what was that? Lorlatinib plus a vaccine plus a different checkpoint inhibitor called CTLA-4. We in the medical committee have been talking to CTLA-4 companies about using their drug with ALK inhibitors. And so we're hoping that we're going to see that come to fruition, especially once the vaccine trials get started. But this is data that supports this idea that you can make ALK tumors sensitive to immunotherapy, and we shouldn't just not include ALK patients because they didn't respond in those initial studies. And here's another example. So CD47 is also a checkpoint. In this case, it's on macrophages, which are a kind of tissue, white blood cell that lives in the tissues. So now this graph is the opposite of the other one. Now, when it goes straight up, it's bad. So across the bottom is days again, but now on the y-axis, it's tumor volume. Obviously, you don't want your tumor to get bigger. So this blue line goes straight up, and that's the control. It got nothing. The purple line got the checkpoint inhibitor by itself. The pink one just got the lorlatinib. And this orange or whatever color it is lorlatinib plus the checkpoint inhibitor. Again, this is in mouse models. We've been talking to a lot of companies to try to get a trial ongoing with CD47 in combination. Most of the CD47 trials in humans have been having a lot of toxicity. So it isn't going to happen immediately, but as soon as we can figure out which of the many CD47s is safe and effective and can be added to a TKI, we want to have a clinical trial because this data is powerful. Again, it shows that just because one checkpoint didn't work, many others could. And so wherever we can, we need to be bringing that to people's attention. Now, why should a company do research in our patient population? And most of you are going to know what I'm going through. But who are ALK positive patients? Well, most are diagnosed 50% or more with stage 4 at diagnosis. That's typical. About a third have brain mets. There's a good amount of data for that. Importantly, the ALK rearrangements or fusions are not usually present along with EGFR or KRAS. And that's important because if you have two drivers like that, it becomes more complicated to treat. And if you're developing a RAS inhibitor, you may or may not want to start with a combination therapy in ALK. So that's an advantage for ALK positive cancers. One of the perplexing things is that PD-1, which remember is something that inhibits T cells, is expressed frequently, but there's rarely T cells in the tumor. So this is commonly called a cold tumor, but we know from the animal studies I just showed you that you can make an ALK positive tumor hot. And that's what we're working on in a number of ways. Young, I showed you in the earlier graph versus EGFR, where a younger population, about half are diagnosed younger than 50. And as we know, most are never smokers. And so when we talk to companies, we say this is a young, otherwise healthy population who's an ideal place to be testing your therapies, as opposed to old people who smoked all their life, who have much more complex medical histories and much more complex tumors. The median survival is getting better. Right now it's 7 years. So we're saying, well, if many of the ALK patients are living 7 years, but they still have tumor on their scans, that's a treatable population because we want to get rid of whatever remains there. That's called the minimal residual disease, that low point in which you have the least amount of cells left after your therapy. So that again is where we've gotten the attention of a lot of companies who are looking at doing immunotherapy in healthy people who have only a small amount of disease for which it's being controlled with their TKI. And I just said that. And lastly, one of the things that makes ALK an advantageous place for companies to do drug development is that you should, and almost everybody is, biopsied when their tumor starts growing. That's really helpful because then you get the information, what is happening biologically in your tumor, and then that company, it's better for them to know when they give their drug and it works or it doesn't work, how to find the right population that can benefit. And that's why it's in red, because when we explain this to companies, they begin to see, oh, wow, look there's 5 really good reasons here for why developing drugs in ALK positive patients could be really helpful to my company. So in the end, and I took out the long summary I have, but just to say, this was the tagline, we believe, ALK positive medical committees, that companies developing lung cancer treatments and diagnostics should have ALK non-small cell lung cancer as a specific and active arm of their research program. Not just well, we'll get to it once we get the drug approved, or it's a really great idea, thank you, ALK positive. No, we think it should be active and it should be specifically delineated for all the reasons I just gave you. And we'll come back to that. So I want to finish on 2 short topics and then Marc will have the rest of the time. we as an organization have decided that we want to support something called Breakthrough Cancer, which is, I'm going to show you in a minute, which is a nonprofit organization already investing millions of dollars in ovarian cancer, glioblastoma, acute myeloid leukemia, and pancreas cancer. And now they want to add ALK. So I'm calling this a breakthrough for ALK patients, because this is going to bring millions of dollars to our most important researchers who are going to be invested in the following. They want to figure out how we can get rid of the minimal residual disease to cure you. That is their goal, and their CEO has articulated that, and we know that, and that's what we want. And we saw this coming together of a common purpose. And so we've had a number of meetings with them, and this is their, oh, and I wanted to just, for those of you that might not be familiar, since this is focused on minimal residual disease, also called MRD, it's the small number of cancer cells left in the body after treatment. And these are the cells that have the potential to come back and cause relapse. So the idea basically is to kill any remaining tumor cells so the cancer won't relapse. And this is a picture of what MRD looks like. They're estimating that 97% of ALK lung cancer patients are in MRD. And you can see here, see the red arrow. You see there's a mass right here. The patient gets treatment, it shrinks down to this size, and that's what their MRD is. And I would imagine there's a number of people who are on this call that know exactly personally how that feels. And then at progression, it starts to grow again. So the idea is to intervene here at this stage so you avoid the progression. And their question, this is right from their slides, why hasn't the problem of minimal residual disease been solved? Well, we don't know why some tumor cells evade. They're going to study that in ALK cancer specifically. And we don't normally biopsy tumor at the residual disease state. That's something that they're going to want to be doing. And they'll be talking to our patients about that, because that's how we find out what is there and what's keeping those tumor cells alive. Trials. We don't have trials specifically at the MRD state. Well, we ALK Positive in the medical committees, we're getting those started. We're not relying on BTC to be our the end of it all. We're pushing ahead with as many companies as possible for patients who are in MRD or progressing. And then finally, they want to do innovation about what kind of technologies, treatments, scans, predictive markers might there be to help us figure out how to solve this cancer at the MRD state. So when I first joined ALK Positive, Marc Muskavitch came to me and he said, Ken, I've got some ideas. And I think we could really make a difference if we started our own internal research group to ask some key questions that could strengthen even further our pitch, our motivational speeches, our efforts to facilitate new drug development in ALK. And this was perfect because when Alice Shaw spoke to all of us at the summit in Philadelphia last year, this is a quote. She said, research focused on defining the biology, the genetics, the immune landscape, and the surfaceome, that's what's on the cell surface of the ALK tumors, will be critical to leveraging new technologies to improve outcomes. So because of that, and because of the funding coming from this patient group, we've been able to put into place some new and creative research initiatives through our medical committees that I think Marc's going to help you see just how it may revolutionize our ability to get new treatments in the development pipeline for patients with ALK lung cancer. So Marc, thank you, and I'll hand it over to you.
All right, thanks, Ken. Thanks for a great summary of many of the directions in which the medical committee is moving. And what I'd like to do is actually take us back a bit, but to explain what the Translational Investigations Group is doing supported by our time and background, members of the committee, and the resources that are been donated by patients, caregivers, families, and other funders to help support research. And also let me mention, with regard to what Ken's been talking about, breakthrough cancer is a marvelous opportunity. And they're going to, we hope, advance a number of initiatives that will help us better understand ALK-positive lung cancer and better treat it, and ultimately, we hope, cure it. But even breakthrough cancer can only work on a few things. They're going to work on probably three major areas of scientific questions, but that leaves a great deal to do, part of which we're doing in the Translational Investigations Group because of the endorsement by the medical committee and the board of directors and the resources from patients and families. So let's step back to the biology. And I want to talk about how we're looking basically at the parts lists for cancer cells, ALK-positive cancer cells, and immune cells in ALK-positive cancer patients, so that we can understand the parts that we can manipulate so as to reduce the division and reduce the viability and increase the death of ALK-positive cancer cells. So let me step back to say that, to remind myself as well, that what happens is our lungs have to renew themselves all the time. So remember, the surfaces of your body are always renewing themselves. Your skin flakes off, your epidermis renews. The inside of your digestive system has to be renewed. The insides of your lungs have to be renewed. And in fact, there's a cell called this AT2 cell, so alpha tango 2 cell, depicted near the bottom of this diagram on the left. And the AT2 cell, thanks, Ken's pointing at it right now, is the source. That cell in a normal lung divides at a slow rate to replace the cells in the alveolar space, where gas exchange occurs, where oxygen moves into capillaries. Those AT2 cells divide at a slow rate, and they replace cells that are damaged because of mechanical deformation, oxygen radicals, and environmental toxins. And so our lungs are always renewing themselves. But what happens in ALK-positive patients is that when those cells are making copies of themselves, they suffer a mutation. And that mutation causes a fusion so that now the ALK-positive kinase, as it's called, is turned on. And if we go to the next slide, what happens is when the ALK kinase is turned on abnormally, it is turned up, it starts signaling to those cells that are normally dividing at a slow rate, divide faster. Because kinases are enzymes that put phosphate molecules onto other proteins often, and sometimes other types of molecules. Those kinases often signals cells to divide. So when the ALK-positive fusion gene occurs because of a mutation in your lung cells, that cell starts to divide too fast, faster than a normal cell. And when cells divide faster, when they copy their DNA, they start to make more mistakes. And so in addition to the fusion change, that mutation in the fusion gene that turns on the kinase, now those cells that are dividing quickly acquire more and more mutations. And the more changes to genes that they experience, the more likely it is that they can become cancerous, ultimately then form a tumor, and then some of those cells can move out and metastasize to your lung, to your lymph nodes, and to other organ systems in your body. So our goal is to kill ALK-positive cancer cells. And actually, when you think about it, there are really three ways that we kill ALK-positive cancer cells. So one thing is we can inhibit, and that's laid out here on the slide, the top one, we inhibit the ALK fusion protein, that kinase. By inhibiting the protein with our TKIs, we stop that kinasing signal, we stop the signal to divide quickly, and division slows. The immune system can catch up. A second option is we can inhibit other proteins that have been turned up because of the ALK kinase signaling in the cancer cells. Because when kinases send cell division signals, they actually turn up many, many genes and many, many signaling pathways in those cells. And we know that from studies of many tens of cancers in tens of thousands of patients. And then the third way we can kill ALK positive cancer cells is we can stimulate the immune system to attack those cells. And that's what the checkpoint inhibitors that Ken was talking about do. They basically take the brakes off the immune system so the immune system can attack our cancer. So on the next slide, we then summarize this. On the left, we inhibit the ALK fusion kinase with TKIs. So we use brigatinib and alectinib, lorlatinib, and some people in trials are now beginning to use NVL-655. Those are targeted kinase inhibitors, TKIs. What the Translational Investigations Group is doing is we are looking to determine the parts lists, the molecular parts, the molecular machines and protein machines that work in our cancer cells. We want to know what machines are turned on and turned up in the ALK positive cancer cells, and what machines are turned up and turned on in the immune cells in ALK positive cancer patients. Because if we can identify those parts, then we can use databases and compilations of proteins for which FDA-approved drugs already exist, and FDA-approved clinical trials for drugs that could affect those targets also exist. And we can identify quickly if a molecular part or a protein or a target is expressed in an ALK positive cancer cell and there's a drug against it, we could combine that drug against this new target, newly defined target, with a TKI, have a new combination therapy, and that might more effectively kill the ALK positive cancer cells. On the other hand, if we understand the machinery of the immune cells, we could basically take the brakes off those immune cells so they'll invade our tumors now and more effectively kill our cancer cells. So how do we figure out the parts list, the machinery of the ALK positive cancer cell and the immune cells in ALK positive cancer patients? To do that, we are actually using technologies that will let us use RNA sequencing, as I'll explain in the next slide, and computational analysis to define the molecular parts of ALK positive cancer cells and of immune cells in ALK positive cancer patients. We can use other information to determine which of those proteins and machines are likely on the surfaces of our cells, our cancer cells and our immune cells, and which are inside our cancer cells and immune cells. So if we go to the next slide, what we're doing is we're using a technique called RNA sequencing. Now, I taught introductory biology for probably 30 years and genetics. I was a professor for 30 years before I joined pharma, and I was in biotech for 10. And so this is sort of like basic science, basic understanding for me. And I'm not going to try to explain RNA sequencing, but let me try to explain that what RNA sequencing does, DNA in our genes codes for RNAs, and those RNAs are converted by machines in our cells into proteins. And so if we can sequence the RNAs in a cell, and we have techniques to do that now, then we can use computers to decode the RNAs that are made in an ALK positive cancer cell. We can figure out how many copies of an RNA for a given protein or target or part are being made. And we can look at those on a cell-by-cell basis in a tumor. So we can take a tumor, break it up into single cells, sequence all of the RNAs in each of those single cells, and then use a computer to tell us which parts are being made in an AT2 cell, which parts are being made in a T cell, which parts are being made in a macrophage, which parts are being made in a natural killer cell. And we can then take this RNA information that nominates a part. A part may be expressed as a protein in a given cell, and we can confirm its expression by looking for the protein in the cell that's been nominated by its RNA sequence. And so what this allows us to do then is take tumors, dissociate them into single cells, sequence their RNAs, use the computer to decode the RNAs, determine which of the 20,000 genes for proteins that exist in our cells are being decoded in the ALK positive cancer cell or in the immune cell. And then we can go to a list an FDA targeted drugs. So if we go to the next slide, this just outlays this. How can we use this parts list to then identify potential new therapies for ALK positive patients? So The first way, the top way there, we could take a combination potentially of a TKI and a drug that is already approved for use in people by the FDA. So FDA approved for administration in people. So if we can take a TKI and combine it with a drug against a different part, a protein that's expressed in an ALK positive cancer cell, now we could put those two drugs together. In fact, if the FDA has approved a drug for use in people, a doctor can write an off-label prescription, and if she wants, she could prescribe us a TKI plus a drug against that part that's in the ALK positive cancer cell, right? We could test that combination therapy if our doctor and we agreed that that's a good idea. Alternatively, we could advocate for the creation of a clinical trial in which we do a clinical trial of the TKI plus that drug. The drug has already been approved in this case for use in humans, so that hurdle's been leapt. But now we just want to try it for a different disease or indication, for our case, ALK positive cancer. A second option is that we can take a TKI and combine it with a drug that is in an FDA-approved clinical trial. So I think most people on the call are aware of the idea that drugs are discovered in laboratories and in companies. They are then approved by the FDA for use in clinical trials, where they're compared to existing therapies, or if they're breakthrough therapies, they're compared to placebo. And then if they are shown to be safe and effective, and more effective than existing drugs, they will be approved and can be used in people. But if we know that FDA has already approved a drug for a clinical trial, we've leapt that hurdle of getting over all of what's called the preclinical work required to get the FDA approval to attempt using that drug in people. And so again, we've accelerated the creation of a therapy potentially because now we know we have something that FDA says we can test in people. It's reasonably safe enough for that. And then the third option is that by looking at the parts list, as Ken mentioned, the medical committee is looking at 70 to 90 biotechs continuously. And we can look at what are called their pipelines. Those tell us what targets they're working on. We can take our parts list of ALK positive cells or immune cells in ALK patients, look at the company's parts list for their pipeline, and say, company X is working on this target, and we know this target is made in ALK positive cancer cells because of our analysis of RNA sequences and their decoding in the computer and their assignment to the cancer cells. That informs then our interest in taking that discussion forward with a given biotech, because if the part is there in the cancer cell, we know their drug against that part may be useful. So if we go, I think, to the next slide, these are the kind of data we get. And so briefly, I'm not going to try to dive into this, but what we've got on the left side are different genes, 13 different genes. And then what we've got horizontally is we've got single cells that have been analyzed in ALK positive tumors, that's what the ALK at the bottom means, in BRAF positive tumors, that's what BRAF means, or in EGFR positive tumors, that's what EGFR means. And when you look at the data this way, what you can see immediately is, for instance, gene G10. You can see, just looking at it, that's in ALK patients. It's not in BRAF patients. It's hardly in EGFR patients. That would be a target that is specific for ALK patients, and a drug against it could be used for ALK patients potentially, right? If the RNA data are confirmed by looking at the protein. If you look at gene G9, you'll see it's present in ALK, it's present in EGFR, and it's not very present in BRAF. That's a drug that could be used for ALK patients and EGFR patients. If you look at genes G6, 7, 8, they're expressed in ALK and BRAF and EGFR patients. Those could be targets for drugs that could work in all of those cancers. And then G1, it looks like it's predominantly expressed in EGFR-driven cancers. So that would be a drug that would be specific potentially for EGFR patients. But these are 13 genes. We are looking at the expression of over 18,000 genes through computational analysis with AirClin company, which is a contract research organization in computational biology working with. The next slide then shows us a different representation. Here, what we do, instead of looking cell by cell, we're looking patient by patient. So across the top are different patients, 10 ALK patients, 22 EGFR patients, 11 healthy individuals. And again, genes are on the left, G1 through 12 in this case, and a red square means the gene is expressed in that patient. And what you can see by looking at this way of visualizing it is the different drivers on the bottom, ALK or EGFR or healthy individuals. And what you can see here, for instance, is that if you look across the row for G2 and G3, you see that genes G2 and G3 are turned on in ALK positive patients and in EGFR positive patients, but they're absent in the AT2 cells in this case. Here we're looking specifically at AT2 cells. The last slide was looking at every cell in the tumor. Here we're looking at just the AT2 cells. So those proteins are present in the AT2 cells of ALK positive and EGFR positive patients, but are not present in normal lung cells. That means they're specific for the cancer cells. We could target them in cancer cells. And similarly, if you look at G6, 7, 8, and 10, you can see that they are on in different patients that have ALK or EGFR drivers, but they're not on in healthy individuals' lungs. So again, those are targets specific for ALK and EGFR tumors. And on the last slide that I have, I just want to say, so where is this taking us? It's taking us exciting and potentially impactful places immediately. So as Ken mentioned on the left, we've looked at these 6 genes in the leftward column. They're the targets for a solid tumor vaccine that Biotech is making with messenger RNA. So just like the COVID vaccine, this is a messenger RNA vaccine against these 6 proteins. We've acquired evidence from RNA suggesting that each of these proteins may be expressed in one or more individuals. And so in fact, in 60 to 70% of the ALK patients samples we've analyzed so far, and that's only 11 so far, patient samples, we find that 60 to 70% of them express one or more of those targets of that vaccine. That has motivated BioNTech to want to create an ALK positive trial arm in a clinical trial. Antibody drug conjugates, we know that TROP2 and HER2 and HER3 are expressed in ALK positive cancer cells. Those are all antibody drug conjugates that are now being trialed in other cancers and could be tested in ALK positive patients because we know those proteins are likely expressed in ALK positive cancer cells. The YAP1 signaling and the beta-catenin signaling pathways are, we think, and scientists think, expressed in persister cells that persist after TKI treatment. The fact that we can say yes and YAP are present in cells in tumors and beta-catenin signaling components are present in those tumor cells suggests that, in fact, we could use the drugs that biotechs are developing for YES1 kinase and for the beta-catenin signaling interaction that we could use. And in fact, in terms of other targeted kinase inhibitors, we know that MET is in fact expressed in many, many ALK positive tumor samples, so that in fact MET inhibitors, as are already being tested in clinical trials, again, are relevant probably to most, if not all, ALK positive patients based on our analysis of RNA sequence expression in ALK positive cancer cells. And finally, the last point I'll make is that when we look in the computer, we can also predict which proteins. Other scientists have predicted that there are roughly 3,000 proteins that are targeted to the surfaces of human cells. We found that at least 650 of those proteins are expressed in ALK positive cancer cells. And so again, we can say these parts may well be on the surfaces of ALK positive cancer cells. They could be targets for new drugs, or if companies come targeting those cell surface proteins, as Alice Shaw mentioned, then we would be interested in talking to them about potentially trialing those drugs in ALK positive patients. So that's some of the investment that you have made that is making possible insights. And what's curious is that though I would think that there would be companies and academic labs looking at this problem in this way, we found that there are not. This is something ALK positive is doing, and nobody else is doing in quite this way, in quite this targeted translational way. So it's something that we are doing as an organization for ourselves and potentially for other cancer patients with oncogene-driven cancers. So with that, I'll hand it back to Ken.
Thank you, Mark. And Yvonne, as the moderator, we're ready to have a discussion with all
Okay,
the
great.
however much time is left. So,
I know, exactly. And I'll ask Mark and Summer to jump in as needed as well. Before we get started, I'm just gonna again remind everyone I'm an ALK positive lung cancer patient. I was diagnosed in August 2021 and thankfully been doing well on my first line treatment, brigatinib. I'm also a board member of ALK Positive Inc. And it's really great to work with this team here because honestly, hopefully you're getting the sense of having Dr. Ken Culver, who joined us about 18 months ago. I think he's really helping to set us apart from some of the other biomarker charities that are out there, and just seeing the way they're thinking about our research, where we are going to be investing our funds, is really, truly exciting. So thank you so much, Ken and Mark, for a great presentation. We'll also be going through the chat and pulling out some questions, but I think one of the things that really came out is, well, maybe Ken, just since you've joined us, I think I can see us pivoting a little bit our research strategy, and I wonder if you could just summarize that. How are you thinking about our research approach? What is it you're trying to achieve right now? If we can start at the high level.
yeah. So when I joined, the medical committee had done a lot of really awesome things, including a number of grants that were really focused on understanding resistance mechanisms, for example. Those are laboratory research projects, and their translation into new treatments could take a while. So what I propose to the larger community is let's develop a portfolio that, of course, has those early research, but it has also some things that could bring results quickly for people who are diagnosed. And I think the medical committee has been working really hard now to find things that are sort of short-term benefit, medium-term, and some that might take longer to get an answer. And I think, what Mark just outlined is a beautiful example of trying to hit the short and medium term benefits by being able to know, well, what is in an ALK cell, and being able then to take that data to companies and say, hey, here's a really good scientific reason that goes along with all the other good reasons why ALK should be part of your development plan.
That's really amazing. And it really comes through in some of the data that Mark was presenting here, how our own medical team is looking at, amazing amounts of data, in a weird way, not to say we're fortunate to have our medical committee with ALK who dedicate their time, but truly it is quite special. In the parts list, some of the things that Mark, you were covering there, you're showing that you're looking at these genes with ALK and EGFR. It sounds like you're using this data to convince the biotech firms to invest in us. Is that the case, what you're doing with this? And is that how we're spending some of our money as well?
Right. So we're spending some of our money to say, these are the parts of an ALK positive cancer cell. And then we'll use that to say to companies that are actually working on that particular part already, we'll say, you should consider, for all the reasons Ken has presented, when you move to trials, putting a trial together that involves ALK positive patients. That's one thing. But then in addition, if we have the ear of a biotech company and we know that they're interested in kinases, for instance, this YES1 kinase that I mentioned very quickly, that may be involved in persister cells, there is a company, Nuvectis, that actually is making very selective inhibitors of YES1. So we're definitely eager to speak with them. And so, this helps us say, yes, we want to spend time building a relationship with Nuvectis so that we could test that particular drug for that part in us.
And Yvonne, let me add something to what Mark said about, so for the group, this is sort of how it worked. I was having a conversation with Christine Lovely. Christine said, you should talk to Nuvectis. I went to the medical committee. Ray Hall reached out to, I think it was Ray or Jeff or both, reached out to Nuvectis. We had a meeting scheduled within a few days. It was at the meeting at the American Cancer Society down at American Associated Cancer Research in San Diego. Colin Barton came, and the two of us sat down. with Nuvectis, and they have data with their YES inhibitor in combination with alectinib that kills alectinib-resistant cell lines. And they're just in a phase 1 trial. So the idea now that biotech companies are looking to define the activity of their drugs in ALK-resistant cell lines before they even have a dose is just an amazing step forward. And it shows the network that our medical committee has within the academic and the biotech community.
That is truly exciting. I saw that press release out the other week, and really truly exciting. And it's wonderful that you're there at these conferences, speaking to everyone and really lobbying for us. I think your pitch in the beginning that we're not rare, and even interesting, Dr. Drilon's comment about rare is not really rare. In lung cancer, just because it is so big. So those are wonderful messages. I wonder, of all the medical companies, biotechs, pharmas, university researchers, everyone you're speaking to, is there anyone that's more promising or seems to be more interested in working with us? What are you hearing?
You're asking me. It's probably a third, and a third rule, Yvonne. There's a third who immediately get exactly ALK. It makes so much sense. We need to do it. And there's I don't want to give out their names because it's not quite finalized yet, but there are a few companies that can't wait to start a trial in our patient population. Then there's that third who, like, you start talking about ALK and their eyes glaze over. And it takes quite a while and a lot of work, if I can get multiple audiences with them, to try to get them the messages I shared with the group tonight. And then there's that group in the middle where they're already committed to EGFR or something, where we're trying to get in and say, can't you kind of add ALK in? It makes sense. There's things that are the same. There's things that are different, like Marc was showing in those track plots. So it's a variety, but we do have some that, of course, Takeda and Pfizer and Genentech are very much engaged because they have a lot of experience with the ALK community. Some of the others that are kind of newcomers, it takes us some work.
That's great. I think it's good that all of them are interested, frankly, because I think we need to look at it from so many different approaches. It's probably not just one way, from exactly what Marc was also speaking about, this kind of looking at attacking ALK from multiple different sources. I was going to go through the chat and also bring up some of the questions that came up throughout the call, and just encourage anyone to add any other questions you might have. In the beginning, Ken, people, I guess when you had the university and institution list, there were a few questions specifically about certain institutions, nothing with UCH Health, or what about MGH, Dana-Farber. So I think people were asking about maybe institutions that they're also familiar with.
Yeah, and they are, and we have no limits. We'll talk to anybody, and MGH and Dana-Farber are on there. So I probably went through the slide a little bit too fast.
Okay.
But yeah, we're no one, we'll talk to anybody. And now that we have all this great data from the project Marc's leading, we have even more reason we want to talk to everybody.
That's great. Wonderful. One of the other questions that came up earlier is about wild type. Could you give an example about what wild type means?
Yeah. So what wild type means is you have a biopsy, they take your tumor, they do a NGS, which is a DNA test that looks at, there's different kinds of NGS tests, but basically it looks to see if there are any genetic drivers in the tumor. They do that. If they see nothing, or they see some things they're not sure about, then they do an RNA sequencing similar to what Marc described. And if they can't find anything there either, no fusions, no mutations, no amplifications, no other rearrangements, then they're labeled as wild type. Okay. And also another thing that goes into that, Yvonne, is that if there's no drug for a subset, then you tend to get treated like the wild type population. So if you find some rare mutation or rare fusion there's no drug for, then you tend to get treated because they're one, they're going to be rare, and two, now finally there are drugs specifically for ROS. And there are drugs specifically for RET that got approved by the FDA that are changing the lives of those oncogenic drivers. So now they have their own little subset.
Great, thank you so much. When you were speaking about the minimal residual disease, someone was asking why can't radiation or surgery be used for MRD?
Yeah, and that is such a really important question. So when the stage 4 cancer patients have think of yourself as having two parts to the disease, what you can see on a scan and what you cannot see on a scan. So if you have a tumor start growing, so if you're metastatic, you have to assume that there are cancer cells in various parts of your body and the TKI is controlling them. And if one starts to grow, you see that, you can irradiate it, but the TKI is controlling all the others. So if we knew where all the tumor cells are, we could probably cure somebody with radiation, but we can't see them. So and even if our circulating DNA is positive, if we can't see them, then we don't know how to focus the radiation or do surgery. So that's really a key limitation. And we in the medical committee, we didn't touch on it tonight, are also trying to convince different diagnostic companies to develop some blood tests that could help predict progression before the scans change, because treating patients with the least amount of cancer in the body is advantageous. So we are working on that. That's a little rougher than the therapeutic companies, but we're trying.
There's also, thank you for that. There's also a bit of a related question, I guess, and maybe this is already somewhat answered, but what PET SUV amounts due to MRD do patients have? Are you even getting, what would you see on the PET, I guess?
I think that's one of the things that the breakthrough cancer is going to do, is try to better define exactly what is MRD, because it's not been something that's really spent a lot of time on. And at MD Anderson in particular, they've been developing a radiomics program where basically they use fancy computer analyses like Marc was describing on X-ray, PET, and CT images to try to look at those features, and based on that, be able to determine what the risks are for progression or metastases or other things. And so I think there's a lot of cool things that are developing out there, and hopefully BTC will be a leader in that for ALK patients.
Let's hope so. Wonderful. And there's also a question here. How can patients convince their doctors to biopsy tumors that are small, under, say, a centimeter, or residual?
Well, I'm not the right one to ask, but of course the biopsy depends on location, not just size. And I think what I'm hoping is that in people's treatment plans, that you have a plan for, at the time of progression, who you're going to go see and who is your second opinion. So if you go to your doctor and they don't want a biopsy, you might want to check with somebody else, because people have different expertise. We heard Matt Quirk in this ALK talk a while back. He's able to biopsy a lot of things that other people can't, because he's developed a very specific expertise. Matt Quirk's part of our patient community. So I think someone else should really answer that question. Marc, what would you say?
I was reading chat, so can you remind me?
Yeah, we were talking about what happens if you can't get a biopsy at progression. What should you do as a patient?
Well, I would think of looking for a second opinion doctor myself. It just is good practice, and the challenge is that it is not yet reflexive practice, and that's the challenge we all face. So I would find an alternative provider to try to get that person to approve the biopsy.
Okay, we also had, I'm sorry, Marc, were you adding anything more?
I was just going to ask whether Summer or others have had any experience with this yet, or Marc have had experience with this that might be useful.
I do hear sometimes they say that it's too small and you have to go the liquid biopsy route, but we all know too with that's all dependent on tumor burden, right? You could be progressing and still and still not show something specific. So it is definitely, I agree, the second opinion. But yeah, I've heard the same even by ALK experts that sometimes it's too small. It's a tricky position.
You know, I think in my case, at least, my progression's always been able to get a needle biopsy or a, you know, something that they have tissue, which I think is still the gold standard. But I think what Ken said recently was just they're trying to develop new tests to get the progression. The other thing too is we can add to this is the CEA markers, the blood markers. And I don't know if Ken or Marc want to comment because I know a lot of doctors don't actually do that. I know CAMAGE is a big proponent of that. So do you have any comments on blood tumor markers?
I don't, but Colin Barton knows. I don't know if Colin's on tonight, but he's much more familiar with it than I think. Maybe Marc, you are.
Right. I mean, Colin definitely, I think Colin has those measured himself. And I mean, I know people with other types of cancer who have had those blood biomarkers assayed to monitor, for instance, ovarian cancer. And I know an individual who that helped probably keep her alive for at least 15 years past diagnosis. So blood biomarkers can help assess the nascent progression, just this very beginning of progression. But again, they're not standard of care yet. So it's a matter of making the case with your oncologist and seeing if they can be put onto your treatment plan, basically. Part of your monitoring.
Thanks for that. Thank you. We also have a question about the section you were talking about, Marc, on the repurposed drugs, using the data that we have, and specifically, is it possible to accelerate a trial once you, I guess, identify that a drug like that is working? Is that what happens? I guess in this case, we do know of gilteritinib, which ALK Positive has been an investor in this trial as well. So is that normally what would happen once you have a drug, you find out this off-label drug, can you move it rather quickly into a clinical trial?
Right. Well, this is closer to Ken's area of expertise, but Ken, I think there's the question of, you know, use off-label versus running through a trial for a registration. So how do those two relate to each other?
Yeah, well, let's use gilteritinib as an example. So gilteritinib is already approved for leukemia. So the big advantage of testing gilteritinib in patients is that you don't have to do any animal studies, as Marc said, and you don't have to figure out the dose. So the way the trial is designed with Dr. Chin at Michigan is you're starting on the approved dose of gilteritinib, and if for some reason ALK patients have more toxicity than leukemia patients, then the dose will go down. It's called a dose de-escalation trial. You couldn't, I don't know, I'm the wrong one to ask whether you could use gilteritinib off label and whether insurance companies would approve it. And without even knowing if it would work, I don't know that. But so I can't really answer you about the off label, but so, you know, immunotherapies are not indicated for stage 4 ALK positive lung cancer, but we know a lot of patients get it at some point in their journey. So there is off-label use of different cancer drugs in patients in the later stages, for sure. So
Right. Well, thank you. Thank you. I don't see any further questions in the chat. Thank you, Sly, for putting in the serum tumor marker numbers there for us. So thank you so much. We had another community member ask if we could share the slides, and we'll certainly be posting the entire chat today on YouTube. And I'm sure Summer, we can make a PDF attachment of the slides also available to that. So really a big thank you to Ken and to Marc for spending your time really breaking this down into such simple, understandable ways for us. And it's really exciting to hear not only where we're investing, but where our medical committee is adding value, really helping to drive the research and to really focus us. So thank you so much for your time. Summer and Marc, did you want to close by saying anything more?
Thank you, Yvonne, for staying up to 1:16 in the morning, London time.
I'm sleeping in tomorrow.
All right, sleep in. And thanks, Marc and Ken, for your expertise. And thanks to all the medical committee members and all the volunteers, because I know you guys just give a tremendous amount of hours to help us and further research. So thank you so much. You guys can unmute. Summer, any other final things, thoughts?
A May calendar will be coming out soon with your save the dates for our upcoming talks. That is it. Thank you so much, everyone, for joining us. And of course, Ken and Mark, Yvonne, also thanks for staying up. So yes, I call our medical committees the wall. You're sleeping. What has happened behind the scenes? So we're so grateful for all of you. It's absolutely mind blowing. We're so lucky to have you on our team. So thank you. Go ahead, everybody.
Thank you, Mark.
Okay.
Thank
Thank
you.
you.
Thanks,
all,
guys.
everyone.
Thank you, everybody.
Everyone.
Bye.
Thank
Mark.
you.
Thank you. Good
Thank
night.
you.
Good
Great.
night.
Thank
Thanks,
you.
everyone. Bye.
Bye. Thank you.